So, dear participants, we are about to start the last lecture of today's schedule. So, today's speaker is Dr. Firoj Khan. He is Principal Scientist and Associate Professor of Academy CSIR. He belongs to Technology Dissemination and Computational Biology Unit, CSIR CMAP Lucknow. He did his post-graduation, prior to joining CMAP, he did his post-graduation from SCAC's School of Pharmacy and Pharmaceutical Sciences, University of California. He did his master's and PhD from UP Technical University, Lucknow, which is now known as AKTU, in the area of biotechnology and bioinformatics. He has been working at CSIR CMAP since 2007. Prior to this, he was part of Institute of Engineering and Technology, UP Technical University. His research specialization include bioinformatics, which has computational biology, component of comparative genomics and transcriptomics. He has an expertise in the area of molecular modeling and cheminformatics, machine learning methods, applications, database and tool development, and RNA sequence data analysis. He has several projects to his credit from UPCST, DST SERB, CSIR BSc, and DST project, DHR Young Scientist project, and DHR EMR project. He has more than 100 publications. So I welcome you, Dr. Firoj Khan, to this Atal-sponsored FDP on drug designing in drug discovery. And I want you to give a lecture on quantitative structure-activity relationship based lead optimization and identification. So now, participants, it's over to Dr. Firoj Khan.
Thank you, Dr. Nidhi, for a nice introduction. And I welcome all the participants online for those who are attending this today's lectures entitled Quantitative Structure Activity Relationship Based Lead Identification and Optimization. I must say thanks to the organizing committee members of Atal FDP on care in drug discovery and design. As you know that this is the a topic related to drug discovery, and drug discovery is basically covering the different components. You see the pipeline of the drug discovery, and you will find four major components. Right from the lead identification and optimization, the next two step is the target identification and its validation. And then after we will go for clinical and clinical studies. So basically, most of the bioinformatics work where computing required more assistant is in the area of lead identification and optimization, and drug discovery, especially the part of target identification part. While the validation of the target is required the wet lab experiments. So we mostly discuss here some of the methods which are very much utilized these days in the identification of potential leads and their putative targets, especially the binding specific targets. And if you see, as the title, today's title, like I've just given some of the part here. So we'll discuss more about lead identification and optimization based on QSAR study. So if you see the definition, and you will understand that we are using the two major components here. One is the chemical structure, and the other is the activity part. Activity means biological activity. In CSR CMAP, I mostly do participate with different chemical molecules, either isolated from the medicinal plants or semi-synthesized by the chemist of the CSR CMAP. So I do analyze these molecules and suggest that how many molecules are more active, and accordingly based on the molecular reason behind the activity, they prioritize the molecules and then they process the same lead to the bioprospection division, where they tested the biological activity through different means. So this is the this component is basically in the middle of the component where we utilize the chemistry as well as the bioactivity evaluation studies. So as the title itself indicating that we are using the quantitative values of the structural activity and the structural properties and make the correlation between these two parts and try to develop the model which can predict or screen the potential molecules. So let's see the definition. If you see the definition as in the slide showing here, QSR is a process by which biological activity is correlated with physiochemical properties and expressed in the form of mathematical relationship. For example, like line equation, which is also called as y is equal to mx plus c. And if you see why we are doing QSR studies, because we identify the potential lead from the plants, but we do not know the reason behind the activity. So we know the molecule is active through in vitro studies or in vivo studies, but we do not know why it is so much active. So the answer, to give why it is so much active, is just we can give the answer through QSR study. So this is the only method through which we can elaborate the chemical structures which are mostly highly active and correlate these properties, chemical properties of these chemical molecules to the biological activity part of the molecule and try to correlate the things and identify the relationship. So this is QSR is basically is also called the classical method of lead identification and optimization. Further, we can see why it is so much important. If you see the US FDA website, US FDA website indicating the importance of usage of different softwares and method. Basically, I highlighted the middle part where it is mentioned they have used QSR is data before approving any drug. So QSR, without QSR data, mostly regulatory body do not approve any drug. So to elaborate the mechanism of action of any molecule, any active molecule, it is necessary to give the reason why it is so much active based on molecular properties or inside hidden properties. So this can only be done through QSR study. There are two types of the QSR studies, 2D QSR studies and the 3D QSR studies. So QSR indicating here is basically uses both the method. And we in the today's presentation, I'm just discussing the 2D QSR with different method. One is statistical method, that is also called as regression method, and the other is machine learning based method. So mostly chemists believe in 2D QSR rather than 3D QSR method. So I mostly use 2D QSR analysis in CSR CMAP. If you see the regulatory body of European group, so they already mention in their website orchestra the requirement for QSR. What are the requirement and how they accept the validated models. So it is internationally recognized method. They have already mentioned there, and it is based on OECD guidelines. OECD guidelines are international guidelines to accept any predictive model. Okay. So just try to go to this website and explore the method and different steps of the QSR modeling. Besides that, you can also explore the OECD guidelines and at what level QSR model acceptable by the regulatory body. You can explore the different parameters and tutorials to OECD guideline, this information is available on the given website. So, brief, I just mentioned that earlier in my presentation that the in silico modeling part, basically the where we use the chemistry related informatic tools or biology related informatic tools, what we call as cheminformatics or bioinformatics. Both the method basically in research lab, we do not like recognize different methods. We consider all this in a single head, what that is called as in silico study. So in silico study basically covers all the in silico methods, computational methods, including the computing one or simulation ones. So if you see lead identification and optimization part, especially the QSR, this part basically comes in between the chemistry and in vitro testing or animal house testing related work. So after like chemistry, mostly the chemists provide the molecule to the in silico study purpose. And after in silico screening and QSR like docking, like MD analysis and ADME analysis and toxicity analysis, we mostly give this result to like bioprospection division where animal house is there. They they test the molecule in vitro or in vivo based and provide the activity information to the concerned scientist or the chemist. And after that, once we get the activity, prominent activity in vivo animal model, as well as in vitro assay, bioassay especially. And in vitro assay, if you see, there are two types of the in vitro assays. One is cell-based assay and the other is target-based assay. So initially we go through with the like cell-based assay and then we go to the target-based assay. And after target-based assay, we got the activity, then we will proceed with the small animals. So we test the molecule on small animals, for example, mice, rat, rabbit, etc. And after that, we go through for formulation and product development. This is the whole cycle for drug discovery and drug design. And CSR, CMAP, and other CDRA lab, especially, another industry, pharmaceutical industry. So now let's see, like, if you see the prior art, how QSR method is developed and why it is so much important these days for analyzing the capability of any chemical molecule as drug liability or not. Pardon? Hello? Is there any issue? Are you able to hear me? Participants?
Yes, sir.
Okay. So if you see the prior art, the QSR method basically established like in 1969 by Hans, who is considered as a professor of chemistry at Pomona College, California, and he is considered as father of computer-assisted molecule design, and also he actually originated the like first time the concept of quantitative structure activity relationship and developed the equation what we call as Hansch equation. In Hansch equation, if you see, he successfully correlated the activity, biological activity with the log p value. Log p is basically called as partition coefficient value, which is a like mimic of octanol water partition coefficient and mimic the biological lipid bilipid layer membrane viability. So he successfully correlated the concentration with the log p and developed the equation, which is a line equation if you see, which is also you can correlate in terms of y is equal to mx plus c. So he plotted this relationship in the form of graph and successfully predict the molecules viability for the human body to cross the blood-brain barrier and plasma cell membrane viability. So log P is basically what it defined. It defined the measure of log P is a measure of drug hydrophobicity. which was selected as a measure of its ability to pass through cell membrane, and the log P value reflects the relative solubility of the drug in octanol, which is considered as lipid bilayer of a cell membrane, and water, the fluid part of the cell and blood. And it successfully correlates that if the log P value is higher, then it is likely chances that it is like highly bioavailable, and it is a good sign to be a good drug. So he formulated like different steps how to develop the QSR model. If you see, there are three major steps in the development. One is training, test data preparation, which is basically experimental data of a small molecule or chemical molecule with their bioactivity data. And out of the training data set or the experimental data set, we divided the set into two parts into the ratio of like 80 to 20%. 80% training part and 20% we kept as a test data set. So this training set basically used for the modeling purpose, model development purpose for them. So we use this training data set in the second step and calculate the different properties, what we call descriptors or the predictors. And there are like 500, 600 properties we normally calculated for each compound and try to correlate these properties with the biological activity and see there is a correlation or not. Once we found the correlation, then we sorted out filter out the different unused properties and selected the best one properties. These best one properties basically correlated in terms of regression, and they have been like analyzed through regression analysis using multiple linear regression analysis and develop the line equation relationship. So the third step is statistical method. For large data set having a correlation, we use normally regression method. For more than 100 to 200 to 300 data set, we normally use machine learning method. Excuse me. So what do you mean by descriptors? Molecular descriptors in QSR have a very prominent role. If you see, these are the chemical properties and numerical in terms of numerical values. And we normally categorize these molecular descriptors in different categories. And these are uncoded structural features of molecules, varying complexity, and also different properties require different time consumption by the computer. So it depends on the property. We normally have like different time scale. If you see the chemical descriptor types, I have divided like in 5 parts, but there could be a more descriptors. But mostly in 2D QSR modeling, we try to develop like a model with different type of the descriptors from different categories. For example, constitutional descriptors, we consider like properties which having a role in developing the constitutional part of the structure, like molecular weight, halogenated atom counts, functional group counts, etc. Similarly, we have divided a group of properties under like electrostatic descriptors, where we mostly cover those properties which having a charges or some kind of areas like surface areas. And the third category is topological descriptors. So we here consider those properties which having a role in topological assignments. So topology related descriptors or properties mostly covers in this area, in this group. And the next group is geometrical descriptors. We mostly also consider those properties which having a role in deciding the geometry of a small molecule. So these are like solvent, accessible surface, isosurface, volume, molecular volume, etc. And the last category is quantum chemical descriptors, where we mostly cover those properties which having a role at quantum level or orbital level. for example, dipole moment, HOMO energy evaluation, dielectric constant, energy, etc. After taking these five types of chemical descriptors of chemical properties, we calculated different properties for different molecules. And after that, we able to develop the model. So different procedure of the QSR modeling are, it is highlighted in this slide. You see, first is selected set of training data where we use normally the drugs or active compounds which having a reported biological activity, and the activity itself may be divided into two types like cell-based activity, in vitro activity, or target-based in vitro activity. Even you can take the in vivo activity also like doses. So it depends on the biological data you have, you can decide the training data set, and accordingly you can develop the model for that specific model. The next step is to calculate the features. Features here means chemical descriptors. So chemical descriptors basically, as I mentioned earlier slide, there are like five types, and we try to calculate as many as possible different properties. And after predicting the different properties, we will able to like detect those properties having a correlation with the biological activity. After getting this information, we develop the matrix or a table of correlation, and this table is called as correlation matrix. Based on the correlation matrix, we prioritize the different features or chemical descriptors and select the important features. Out of like 500, we select almost 100 or 80 or like 60 descriptors. Most important which having high correlation. So it's a very tedious job, you can but do it. And after that, divide the training data set into two subgroups. Like 80% part you have to select under training set, and 20% part you have to select under test data set. So 80% part basically used for model building, and 20% test data set will be used for evaluation purpose later on after development of the QSR model. So the next step we have build the QSR model. So what are the important features you have to keep in mind? Basically, you have to develop the model using multiple linear regression or partial least square method or machine learning method. Any method you can. You have to like it depending on the training data set. If you have a large training data set, more than 100 or 200 or 300 or 500 or thousands, to I suggest you can use machine learning method like SVM or like other method, ANN method, for example. It's up to you, like see the performance and see the evaluation part. Based on that, you select the method. So what the important feature you have to keep in mind while developing the QSR model, that the regression coefficient should be more than or equal to 0.6. So 60% model accuracy should be there. If you are able to develop the relationship more than 60%, then it is a likely chance that you have developed a good model. So this model can be like have a property where you can like able to publish it in any good journal, and but it takes time actually. The next step after developing a model with 60% accuracy or more than 60% accuracy is the model validation part. So here model is basically a mathematical equation. You see, this is a line equation, y is equal to mx plus c. This equation in terms of properties basically called as QSR model equation. So this model equation covers the training data set, and the next step is validation. After developing a model using the training data set, the next step is to check the prediction of biological activity through this model, and later on compare with the results of the test data set and see the differences in prediction. So lower the differences, higher the chances that you have developed a good model. As I mentioned in the like this part here regression coefficient is referred by QSAR or RCB square. RCB means regression cross validation square is should be like more than equal to 0.5. So this is a criteria for selection of good model. And the last part is to after developing a model validation part, now you are able to test the model in any molecule. You you can predict the biological activity of any unknown molecule. These unknown molecule basically considered as query set molecule or query set data, and this way you can screen number of molecules. But the important part you should have kept in mind that uh, whatever the molecule is selected for analysis, it should follow the applicability domain. Applicability domain is basically the criteria where we select the unknown molecule whether it's able to cross the uh, fit the model or not. So if the molecule is basically out of the range of the model training data set model, then it is likely chance it will give the false prediction. So to avoid the false prediction, we normally utilize the applicability domain area where we see the properties of the query set molecule with the training data set molecules. If they are in the within the range, then it is acceptable. Otherwise, it should be avoided. So after developing the model, what are the advantages of the QSR? You see, there are lots of advantages I mentioned here, but there are disadvantages also. So be careful for utilizing any QSR modeling tool. Number one is understanding of the effect of the structure on activity. We can understand this through QSR study. Also, at the same time, we can get the idea to make prediction of any biological activity using some training data set. So depending on the chemical features, we can predict the biological activity. This is a good part without knowing the experimental activity. So without having a experimental part data, you can evaluate any molecule and able to predict the biological activity. So this way we can sort out or prioritize the molecules for in vitro testing. And this way we save the wet lab economy, time, and the life of the small animals. The next point, it is mentioned that to help understand interaction between functional groups in the molecules of greatest activity with those of their targets. At the same time, we can able to understand what part of the small molecules or which property basically regulates the biological activity on a specific target. So this can be answered through QSR modeling. Now see the application part of the QSR. If you see the application part, there are so many advantages and applications. The one is this model basically used in biological activity prediction, prediction of chemical toxicity, prediction of environmental activity or toxicity, prediction of molecular properties, save the wet lab economy, time spent on biological testing, at the same time save the life of small animals used under preclinical experiments. So these are some of the applications. Now let's see the disadvantages of QSR. If you see, if the training data set is not good enough, then the model built will be a false model basically. It will give the false results. A false correlated model may arise with a lot of noisy data. So this is a disadvantage that we don't have any criteria to select the best one training data set. It's up to the depend on the researcher or the group who able to decide the training data set size. Otherwise, it's mostly give the false positive results. So it requires lots of iterations and lots of careful data selection. Data collected may not reflect the complete property, and this way, the results it will result cannot be used for confidently to predict the biological activity of unknown compounds. So this is a very important drawback of the QSR model. Because in natural compounds, if you see, like if you see the drugs for a specific disease, there are not so much drugs available, like in Corona case. So how you can develop the QSR model? Without any training data set, you cannot develop the model. So that's why in this case, where we don't have any drug molecules, we cannot develop the model. So this is the limitation. And the next limitation is selection of the features or the properties. Features may not be reliable as well. This is particularly serious for 3D features because 3D structures of ligands binding to receptor may not be available. So this is again a very important drawback. And there are many successful applications, but do not expect QSR work all time. In the last slide, I just mentioned here that QSR is not the guarantee that you can get a good lead. Depending on the team and the expertise, whether you are able to correlate the biological activity with the help of the properties or not, it will depend on the knowledge of the particular group and the selection of the prior established data or reported data. If you are using only the databases, small molecule databases or bioassay databases of NCBI or EMBL, a drug bank, etc., they will not give the good QSR model. You cannot able to get the QSR model. So you have to at the same time you have to use the publications, see the publications, and go through with the molecules, what they have reported, and collect the some of the data from literature also, and some of the from databases also, and some data from wet lab also. So in CMAP, I used mostly 3-4 combinations. Like I use chemist data as well as publication data as well as databases data, and altogether they are able to make a good database for me. And we utilize this database for development of QSR model. So you can do the same thing in your lab and try to develop a model. Now let's see the example of QSR modeling and how to know that whether the model is good enough or bad enough. Let's see two examples here. Before that, I just like to mention that QSR is very traditional approach, and there are in the past there are a number of drugs already released with the help of QSR method, and some of the example I've shown here, which are commercially available drugs, which are developed through QSR, classical QSR method. For example, fluvoxamine and bromobutane, norfloxacin, lomrazine, metconazole. They are already means day back, like year back, they have been established through QSR model. Now let's see the example one where we use the multiple linear regression method and partial least square method to develop the relationship for galvanic acid derivatives for immunomodulatory activity as well as anti-inflammatory activity. We got some of the experimental data from the CMAP animal house for some of the galvanic acid derivatives. So we correlated the earlier reported known drug as well as reported active molecules. If you see the table on the top, I just listed some of the drug, not all. And these drugs basically called as training data set, and their biological activity in terms of lethal dose 50, LD50 in mg per kg. And then we, in the second column, if you see, we normalized it in the form of logarithm data, and normalized data basically used for model development purpose, and we build the model. The model equation given below, you see in the end of the next slide, QSR model equation is there, where I mentioned five, six properties which are highly correlated with the activity, because we got the R square or regression coefficient value equal to 0.99, 99%. We got the correlation. And similarly, the cross validation coefficient value is 96%. So we got these 4-5 properties dipole moment, steric energy, group count, lambda, and max UV visible, molar refractivity. These five properties basically controlling the in vivo activity in terms of, it is mentioned here in C, in terms of LD50, and we predicted the biological activity of unknown molecules, which are designated as G1, G2, G3, and G4 in the table. And you see the red color G3 molecule, which is we have predicted their biological activity as 1.908, and which we found in wet lab the same. So we prioritize the molecule based on the QSAR model. We screen the molecules and predict the activity. The graph indicating the linear relationship between predicted and experimental log LD50 value, which shows the high correlation between the properties and the chemical biological activity. So this is a one example, and it is already published. You can explore the internet and try to find out this type of the paper with the title search. And in the second example, if you see, this is example number 2, where we use the OECD guideline based modeling, where the training site is 28 and the test site molecule, we use the 6 molecule as a test molecule, and we find out 4 properties mentioned here as SSN count, nitrogen count, and quadruple to NCN1 and Chi-B5 chain. These 4 properties basically correlated well with the biological activity, and their equation we have already mentioned on the left hand side, top side, and we predicted the activity of unknown compound, and we found experimental and predicted value almost similar after the experimental evaluation. So this means this model is basically quite well elaborated with correlated with the biological activity, as it is seen in the graph. If you see the graph in the middle, red color dot indicating the training data set and blue color dot indicating the test set molecules. Both lie in the linear fashion and in diagonal way, and they are distributed right from 0 to 2.6 in x-axis. So they are highly correlated, it means, and you see the R square value is 0.89, 89% correlated properties, while the Q square, which is cross validation square, cross validation regression coefficient, which is 85%, 0.85. So normally Q square value is basically lower than the R square. So this is a good indication that the model is highly correlated. At the same time, you see the contribution plot out of like 4 properties, how many properties are positively correlated with the biological activity and how many negatively correlated with the biological activity. You see 2 properties uh, below the 0 value, uh, quadruple and TCN1, they lie within the negative region. So it means they are negatively correlated, while the first property SSN and nitrogen count and Chi-V5 chain is above the 0 value, 0 axis and y axis, you see. So they are positively correlated properties. So it means if you increase these properties, biological activity will increase, while these other 2 properties have an inverse relationship. Now let's see on the plot one. If you see the training set mapping plot and test set mapping plot, both are showing the overlap, 100% overlap with the red color line and blue color line. Red color line indicating the actual data line, and the blue color is basically the predicted line, which is given by the model, QSAR model. So you see there is overlap, 100% overlap, and in both the cases. So this model is best fit basically. So this way we can evaluate that the model is good enough, and we can select it for publication purpose or screening purpose. Now let's see the other example where we can see that the QSR model equation properties are good enough. But if you see the graph, training set and test set graph, training set plot is basically somehow overlapping, but not 100% overlap. They have some gaps. But if you see the test set data, so there is a large gap basically. So this large gap basically indicating this model is unfit. So you can reject this model, although the regression coefficient value is good enough, and they are linearly fashionably arranged in the plot, and QSAR value is also good enough, 0.79. So even the properties are good enough. The QSR property should be like not more than 5. So there are like 4 properties there, and the training set data is basically more than 20. So it is there. Test set data is also 7 good enough for evaluation. So this model is not good enough. So we have rejected this model. Now let's see the example of machine learning application. When we have a large data set, as I mentioned in the earlier slide, that if you have a large data set, you can use the machine learning method. In this case, in this slide, you see the title is cluster based. We use the clustering as well as SVR. SVR here is a machine learning method called as support vector regression. So we use the SVR and clustering both in QSR modeling for high throughput screening. STS means high throughput screening records. Screening of anti-cancer leads against human breast cancer cell line MCF7. So we use the breast cancer cell line activity data to correlate with the chemical properties and try to develop the model using the SVR approach as well as clustering approach. So in this, if you see, total we use 2291 compound from NCI database. All have biological activity on breast cancer cell line MCF7. So there are like high correlation. If you see the comparison between experimental and predicted log value, we developed the model with the accuracy of 88% and Q star is 87%. And we got the almost similar activity in the wet lab. So if you see what we have done, we have taken 2291 compounds and classified in different groups. So each group basically have a representative centroid molecule. As shown in the figure, you see there are lots of, in the cladogram you see lots of molecule are mentioned in the end of the branch, and the overall central molecule is basically having the structure which is mentioned on the top of the tree. So this way we able to like classify the whole biological molecules, that there is structural diversity, 8 types of the different structural diversity within the biological active molecules. So structurally they are different, but biological activity wise they are similar. So we classify these molecules in 8 groups. These groups are called as clusters. Cluster 1, cluster 2, cluster 3, cluster 4, cluster 5, and so on. You see each cluster having a different chemical structure, representative structure, which is also called a centroid molecule. So you see there is a structural diversity. Each group has different type of structure. But the good thing is that all structures have same biological activity. They all are inhibiting the breast cancer cell line in the wet lab. So it means the structural similarity is not necessary criteria. There should be more properties hidden within the structural part which need to be explored. So we explored these properties one by one. For each compound, we explored 700 properties. And likewise, we calculated for 2291 compounds different properties and correlated these properties and classify these drugs molecules in 8 clusters and develop the model for each cluster. You see in this table I mentioned here that the R square value for each cluster is different, because each cluster has different chemical structure, and chemical structure if change, property change. So we choose the best one. We have like option to choose the best one property. So we cluster the molecules, and based on this performance, we select the ultimately the final model for predicting the biological activity. So this is the plot for correlation regression plot. We made linear. You see, linear regression is there, and in this presentation, you see the structural diversity and their predicted value from each model, and we found experimental similarity in each case. Now let's see the another example where we use the machine learning method and we simulate the biological activity keeping in mind the time taken during the biological activity evaluation. For this, we simulated in silico assay model plot for two molecules, Inothra A and Inothra B, where within the range of concentration gradient 0.01 to 500 millimolar and micromolar and four different time intervals: 24 hours, 48 hours, 72 hours, and 96 hours. So we model this variation of the activity and taking the picture into the form of mathematical impression and plotted in the graph. You see, in the graph we have to capture the picture of in silico model as well as experimental model, and we try to develop the regression model, and after that we are able to understand at what level there is a difference in the activity. So accordingly we are able to identify the chemical part where the biological activity changes based on the testing on a specific cancer cell line. This is further evaluated by entity bioassay, and we found that both the molecules showed the good activity, and these two molecules are from plant Inothra, biennis, roots. So these are already reported, and if you can see the detail in it is already published in PLOS One. You can explore this paper. Another example, if you see, you can develop the model for, we have developed the model for uracilic acid. You can follow the same route and try to develop the model like this and synthesize the molecule. In this model, we suggest the chemist to choose the best molecule for synthesizing the optimized list. And based on the optimization, they can further change the side chain variation and do predict the same thing. And if the biological activity improve, then we select the biological molecule. So whole exercise already mentioned in the published paper, you can explore in Journal of Molecular Modeling, and with the author name of Kumal Kalani. She actually done the work actually for uracilic acid analogs. Likewise, we have developed free QSAR anti-cancer model based on machine learning. You can also do the same thing. They are already published. Try to explore more detail from literature, and at the same time, you can develop the QSR model. So in CMAP, we have number of QSR models, and we normally use in offline for a screening of molecules from different chemical labs. So we have medicinal chemistry lab, we have analytical chemistry lab, we have sample synthetic lab. So we try to give the answer to QSR model. So right now we have, I've listed some of the QSR model here. We have a QSR model for glyceric acid derivatives. We have a QSR model for uracilic acid derivatives for anti-cancer activity. And we have also the QSR model for camptothecin derivatives for DNA topoisomerase inhibitors identification. Similarly, we have gravifoliol QSR model and indole chalcone and foxcholine derivatives also. So these are all published model you can explore, and if you want to screen your molecule, you can explore and take advantage of the CSIR lab and try to predict the biological activity and then get tested in the wet lab. Similarly, we developed the QSR model for antibacterial, antimalarial, antifilarial, and anti-inflammatory uh, molecules from plant origin. So we have listed details here. Just try to go through it and explore these data from the internet. already most of the like work is published. You can go and see the detail of like QSR, docking and admit studies data, which help the chemist as well as the biologist to come out with the potential lead, having a scientific data with mechanism revealed. So like this, so far in CMAP, I have identified potential lead with glyceric acid derivatives and ursolic acid derivatives, chalcone derivatives for different diseases. That is mentioned here: anti-cancer, anti-filarial, antibacterial, anti-inflammatory, lignans, diagenin, brevifolone, camptothecin, tetralone, etc. And presently, there are like already going on work uh, on anti-cancer going on. We will publish soon. And in this slide, just I mentioned the like in silico assay we have developed for STAT3 inhibitors. You can also develop through using the QSR method and publish your data. And if you have a, like a QSR data, like if you get the information from training data, which is very specific to target. So target-based bioactivity model you rarely find in the internet or any other sources. Mostly cell-based or cell line-based data you easily get online in the databases or in the literature. But target-based assay information for a particular chemical series is very tough. So we luckily found the data on target-specific biological activity and we developed the model. So this is the model we have developed on STAT3, which is a target for anti-cancer activity. And we identify 17 phytomolecules, plant-based molecules. They showed good biological activity out of 2,837 molecules using the CAT database. So likewise, here in this model, we have used the Gomberg's function, which is a mathematical modeling method other than regression. This is again a machine learning method, Gomberg's function. You can use this method also and try to develop the uh, model. So depending on the like training data set, you can develop the model. So if you have very low data set, you can use mathematical function like Gomberg's function. Or if you have 100 up to molecules, you can use regression data set. For more than 100, you can use machine learning method like SVM, SVR, ANN, like that, or clustering. You can use that. So I listed in there the topmost molecules which having the activity against the STAT3 target of cancer. And these are the like 17 plant molecules which I've shown here with IC50 inhibitory concentration 50% is ranges from like 5 micromolar to 12 micromolar range or 16 micromolar range. So the like biological activity with like up to 10 micromolar consider is very good and lower than 10 is very considered very significant. So more than 20 micromolar activity seems very mild or no activity. So keep in mind while predicting the biological activity through QSR study. So this is a brief example where we use the Gomberg's function. Gomberg's function basically consider the time based on some biological function. Here biological function is activity. We consider the activity in terms of time. So these two parameters considered parallelly. We used, which is basically a common feature in biological assay. We use and mimic the same thing through this method and develop the model. So this is a whole summary of the like developing a model for in vitro assay. So before like going for in vitro assay or spending so much money on like small molecules because chemists provide lots of molecules, but how to select the best one? See, so normally chemists provide all the data to in silico lab, and we normally screen the chemical molecules and prioritize the molecules. Out of like 100 molecules, we provide like 4 or 5 molecules for testing. So this is a like QSR modeling approach is like a screening lab to cut down the large molecules which are like mostly randomly provided by the chemist to the biological activity. So this is a very good method to cut down the cost of wet lab. If you see like in this diagram, When we get the biological activity, we explore the molecule in admit properties of FDA approved software admit predictor through Topkit software. And this diagram showing that the molecule within the circle are basically more bioavailable than other molecules. So bioavailability means crossing the biological membrane from intestinal membrane and reach to the blood using the like cross the bilipid layer membrane of the cell. So this parameter is very much required before testing on animal, small animals or clinical trials. So this is a pharmacokinetics parameter evaluation plot where we compare the known drug with the target molecules which we have taken here. So if you see through QSAR model, we identify most of the active molecules which lie within the circle, within the different circles. Each circle indicated by different properties like admit properties called blue, absorption 95% by red color line, and absorption 99% by green color line, and purple color line indicating blood-brain barrier crossing line. And similarly, 99% blood-brain barrier line is indicated by cyan color. So you see, most of the molecules lie within the center are basically good target molecule and maybe a good drug for future drug development. So we normally use these drug in formulation, herbal formulation, and either we provide the same to the pharmaceutical companies or to the technology transfer team members. Excuse me. Now let's see the another cluster-based support vector regression method where we used different clusters and we developed the model, and here we briefly summarize the like method to identify the different molecules. And I have already explained earlier the same thing, but this diagram indicating the flow chart of the method. So you can explore that we randomly, if somebody provides a molecule, how can you like identify which which QSR model is good enough to predict the biological activity? So the one option is to like do the pre-screening. If you see the first option of the diagram indicated by dotted red color lines, so pre-screening method basically helps a lot to screen at the start that randomly if somebody provided a molecule, just try to compare your molecule with the all these center cluster molecules, and those cluster is showing the good similarity or matching more than 60% similarity with the structure. We normally select that cluster. Like example in this diagram, we use cluster number 7. So we got the molecule randomly on the top. It is basically a within a light molecule, a plant molecule. We compared randomly with the model cluster, and we got the match with the cluster number 7. So we put this molecule to the cluster number 7 and predict the biological activity, and we got the activity. And you see, we have the post QSR modeling option. We have 4 options. Either the molecule is inactive, or less active, or mild active, or are active. So you see, the last option we found the biological activity value, which comes under the active region, and experimentally we got the same results. You see the experimental log GI50 values, growth inhibition. GI means growth inhibition. So experimental log GI50 value is minus 6.73, and predicted log GI50 value is also the same minus, that is minus 76.7. So you see the importance of the QSR modeling and feature selection. You can easily predict the biological activity of any unknown molecule before going to the wet lab or spending so much of money in vitro assay or in vivo activity evaluation, or even or before clinical trials. So you must screen your molecule through QSR before doing any wet lab experiment. This is the best method so far we have analyzed. For your reference, I just mentioned some of the snapshot of the published paper from CMAP or my lab. Just try to explore, and I suggest to try to understand the importance of the different method of in silico study or identification of drug molecules or drug like molecules. We cannot say drug because drug molecule, when we say, when it is like after clinical trials to phase 2 or tested positively on patients, we normally release the molecules. Once we release the molecules or regulatory body approve the molecule, then it is called as the same molecule called as drug molecule. Otherwise, we normally use the term lead molecules. So under lab, especially the lab work, lead identification, we normally use initially the term bioactive molecule or active molecule. And after preclinical trials on small animals, we use the word lead, and after clinical trials, we use the word drug molecule. So this is a use of the terminology for the same molecule. So in this slide, I just mentioned the anti-tubercular activity of glycyrrhiza glabra molecules. So you see, we have identified the uh, bioactivity guided isolation of glycyrrhiza roots and resulted glycidic acid majorly molecule which considered as an anti-tubercular agent based on the activity. So we predicted 2 3 molecules here, GA3 and GA8 as a good active molecules, and it is basically found very prominent molecule against polymerase RNA polymerase subunit RPOB target of Mycobacterium tuberculosis. Uh, so we normally published this work and also provided the same to the repository, chemical repository of the CMAP, and it is under formulation process and soon maybe transferred to the some of the pharmaceutical company or to some uh, herbal formulation company. This is another molecule where I work with the team of the chemist and the biological activity evaluation people in CMAP, where we identify as the molecule which basically showed the antiplasmodial activity using the molecule of oxalic acid, which is a triterpene molecule, and we got good QSR model. We developed the QSR model with 96% prediction accuracy. In this, we use the multilinear regression method, which is a very important method you based on load training data set. And for validation, we use the leave one out method or LU method for validation, and we found that it has a good activity on prediction accuracy. And later on, we predicted the in silico admit properties and toxicity properties and rule of five data, and cross-validated through MTT bioassay and found that most of the molecules lie within the selected range. This is a one way. For more detail, you can explore the publication, which is published in RSC Advances in 2015. You can try to explore the paper. Now, this slide basically showing the tool which we have developed in CMAP. It is a repository tool for offline work. The CMAP scientist and biological activity people or the chemist, where we in this basically QLead is a tool for submitting the biological
Hello, dear participants. Due to a power cut, there is a problem for 1 or 2 minutes. As soon as the generators would be on, Dr. Firoj Khan will be sharing his presentation.
Hello,
Yes,
Firoj sir, may I ask one question?
actually, he is offline. There is some power cut in his institute. So just
Okay,
wait for
okay,
two,
ma'am.
3 minutes.
Okay, okay, ma'am. Okay.
Sir, I think power is back. Now we can see you. Sir, voice is not coming. Your mic is muted.
Hello, are
Yes,
you able to
sir.
hear me?
Now we can hear you.
Okay, okay. Sorry for the interruption. So, if you in this slide, if you see all the QSR models of our develop, it is already listed and submitted, and you can use. We normally use these model for screening of the small molecules. If you see in detail, you can also visit the same page online using the search on Google by searching Q lead. In this page, if you go into the model page, you will see a brief description and a QSR equation. And below that, if the data model is published, it is the reference here. And hello, participant, are you able to hear me? And sorry
Yes,
for
sir,
the
we can
interruption.
hear you. We can hear you.
Okay, okay. Thank you so much. Actually, somehow I don't know why it is happening. Uh, let's start. So in this, if you see lots of small molecules which are lesser than 300 molecular weights, which are considered as part of the family of triterpenes, monoterpenes basically, and sesquiterpene, they are basically aromatic in nature. They are like mostly considered as aroma molecules. So they also have lots of biological activity. So if you want to try to, if you are like planning to do work in aroma related or cosmetic industry related QSR modeling work, try to explore this database. We have developed aroma molecule database. It is available.
Sir, voice is not audible. Sir, not able to hear anything.
Hello, sir, your voice is not coming.
Sir, not audible, sir.
Hello, are you able to hear me?
Yes.
Sir,
Okay. So this slide just try to see we have listed the bioavailable parameters, different parameters where you can assess the molecule whether it is likely to be a good for a drug molecule or not. So there are like drug likeness score parameters we normally analyze and different toxicity parameters we analyze as listed in the table below. In this example, you see, once we do the ADME analysis, last part is toxicity result. So we normally compare the data with the control drug molecule. See, on the last column, doxorubicin is a known anti-cancer drug. We compare the different biological properties related with toxicity with the other molecules. As mentioned, it is X19, X44, X45, and X49. These are the xanthone derivatives molecules which consider very active in the lab for anti-cancer activity. And we compare the toxicity data, especially the mutagenicity, anti-cancer activity related uh, properties different, and skin irritation, mutagenicity, like that, and reproductive toxicity. So you can also do the same thing here. After like analyzing the data through QSR, you can do the EDME analysis as well as toxicity analysis at the end. And then you can do the molecular docking with the target molecule, target protein molecule, and see the interaction, whether they have lower binding affinity energy or not. If their binding affinity is lower, binding score is lower, then it's indicate that it has a good binding over the binding target. So you can explore the same thing, and also at the same time you can predict the different targets, because one molecule may have some property to target different targets. So at the same time you can explore the off target also through target mapping exercise as listed in the network diagram. So this is the work which we have already exercised, and it is already patented, and the formulation is already uh, given to the pharmaceutical company. It is a anti-socratic formulation to act against skin cancer related disease. Likewise, you can do 3D QSAR modeling using the pharmacophore mapping and docking exercise. I'm not going in detail, but I just try to show the snapshot of the same here. We already done, and it is a good idea to reflect the biological activity or mechanism of the small molecule using the steric energy or electronic derivatives parameters. If you see the FDA approved drug, most of the like recent drug approved as listed here are basically monoclonal antibodies and protein kinase inhibitors. So you see these are all are basically identified through structure-based drug design, computational drug discovery scheme. All have like computer CAD related experiments. So once they analyze the experimental part through computational studies, then they finally design the molecule, and which is already in the market with different marketed name, brand name, basically. I'm not going in detail much. Yes, just for your example purpose, I just listed the name of the drug. Just try to explore each and every drug. They have somehow used the computational method at different stages of the development, and different pharmaceutical company I have already listed here in detail. If you go, Novartis, ISEC, and even the Bristol Myers, Gentech, etc. They all have using a computational application. They have a very good computational biology lab there. They normally use different QSR method, ADME analysis method, MD simulation, docking, rule of 5 parameter, biability parameters, PKPD analysis, etc. So you can explore the different method in CAD, and this workshop, I think, organized by Dr. Nidhi at IIIT Allahabad, it's a very good exercise to explore and go through the detail of the drug discovery stages. And I hope you will be benefited with this training program at the end, and try to set expertise in some of the areas of the computational experiment. With this, I'm just completing my presentation. Thank you for your patience and listening. And if you have any comment or feedback, please give your comments. Thank you. Thank you, Dr. Nidhi. Please proceed.
Thank you, Dr. Firoj Khan. Now to the audiences, if you have any questions from Dr. Firoj Khan, he would be more than happy to answer you. So kindly put your questions to him directly.
Hello.
Yes.
Firoj sir, myself, Dr. Rambabu Tripathi. I am working here in Ramesh Institute in Pharmacy Department. My question is that what is the criteria for the selection of QSAR series? When you
Basically,
perform the QSAR, number of series are there. So what is the
yeah,
basic criteria to select the QSAR series?
it depends on like what you're like going to test on. If your objective to test on some indolone derivatives, for example, indolone derivatives you have, and planning to predict the biological activity or like in vitro activity you're planning to do in indolone derivatives, then you have to develop the training set structures having the similarity with the indolone structures. So you must have at least compare the structure using the SMILES method. Take the SMILES of the structures. SMILES is a form of linear structure arrangement where we compare the one structure with the other structure, just like blast analysis, and see the similarity. If you have more than 50% similarity with the structure, you can build the training set molecule. So otherwise, you can very specifically identify, do some prior studies work on indolone derivatives of one chemical series, and develop the training set model based on what you are like planning to do on.
நீல தூங்கிட்டு இருக்கிறேங்கிற தம்பி என்னடா என்னடாச்சு யாருடா அடிச்சா கண்ணத்துல கை பதிஞ்சிருக்குதுடா கண்ணு குட்டிக்கு என்னடாச்சு
Any other participant? I hope you got the point.
Yes, sir. My next question is that, sir.
You can contact me later on. Okay, if you require any help, I just help you.
One person is asking about the role of artificial intelligence and machine learning tools for QSAR modeling.
Okay. So as I said, once you have a large data set, for example, you see the AMBIL, Campbell database or EMBL database or the drug bank database. If you have more than like 200, 300, 1000, 2000 molecules with biological activity, experimental biological activity. Experimental means in vitro or in vivo.
So next question is
Basically machine. Yeah.
which
Please.
properties you should consider for clustering?
Okay. For clustering, basically depend on like, I suggest your feature selection is another criteria. There are two things. One is the selection of chemical properties and other is selection of the molecules, training set molecules. So for chemical feature selection, we normally do the principal component analysis. So randomly you select best properties and try to make the PC component analysis and find out the most potential properties. Otherwise, other method is correlation coefficient matrix. So you can try to build the correlation with the biological activity of molecule with the properties and find out the correlation which have more than 0.5 correlation value and select those properties for feature selection. So this is the two properties method normally we used in lab.
Okay, sir. Then one question is, what are the criteria for? I really don't understand. What are the criteria to focus for selection of PDB?
PGP. Ma'am, please repeat the question actually.
Question is quite not clear. I would request Deepika to Dipali Nahar to
Okay.
her question because it's not clear to me. She writes how to select PDB.
I don't understand what the meaning.
The question is itself not clear.
Yeah.
Okay, so moving on to the next question. Tools available for selected ligand and targeted QSAR studies.
Okay. There are like so many software which are commercially available, freely available, very rare, but you can explore on Google search. But I mostly use Cypress, Discovery Studio, Sibyl, Schrodinger. They are have a good V-Live software even also. And machine learning basically are open source available tools. So you have to compile all the different components in Linux. We normally use the Linux platform, Ubuntu. And on this Ubuntu platform, we normally use different Linux binaries for building the model. For example, R Studio, you can use the R language, Python, you can use. They have different libraries. You just select it. Like for example, ggplot. ggplot, you can use this tool for developing different kind of plots related with regression analysis and other statistical tool also. So you can explore the different tools of open source available. There are so many available.
Okay. So I think the questions have in my chat box. I cannot see any more questions. So we are almost at the end of the session. Thank you, Dr. Firoz, for such a wonderful talk. And it was quite enlightening on the topic of QSAR. I think my participants have learned a lot from you. And your email is
Hello,
available
madam.
on the
Sorry
screen.
to interrupt.
Yes.
Can I ask a question, madam?
Okay, okay.
Yes, ma'am. By using this CMAP database, can we run our QSAR studies for our own design molecules, sir?
Yeah, you can download the tool,
Yes, sir.
small molecule database, and you can try to build the model. And even you can use the models available because
Yes,
in
yes,
QSAR,
sir.
if you see, there are so many QSAR model. You have to
Yes,
just
sir.
register there and predict the activity for different series, chemical series, because there are different chemical models, chemical series based model there are. So you have
Okay,
to
sir.
choose a different model based on your chemical series
Okay, okay.
and biological activity also.
Okay, okay, sir. Once after completing our study, can we go for publication in case
Sure.
of a few,
Yeah, you
few
can go
licensing
for
matters and all are there?
publication.
Yeah, okay,
Sure.
okay, sir. Okay. Okay, sir. Thank you, sir.
Thank you.
Thank you. That's it, sir. Thank you.
Okay, so are there any more questions? So I think we are at the end of the session. I thank my speaker, Dr. Firoj Khan, for such a wonderful talk. And I thank him for satisfying all your queries as well. I hope this session has aroused some interest about QSAR in the participants. Thank you, Dr. Firoj Khan.
Thank you, Dr. Nidhi. Thank you for giving me opportunity, and thank you all the participants. Happy learning and learn more. Best.
Okay, thank you, sir. So participants, so this is the end of today's schedule. So tomorrow we'll be meeting at 10 o'clock with our next talk. And this is to inform you in the tomorrow's session, we will be having hands-on experience as well. Okay, so we'll meet tomorrow at 10 o'clock.